Okay, good afternoon, everyone. We are going to start kind of in a way based on the syllabus. Okay. Until now, I have covered some different techniques in the analytical perspective. In today's and in the following 2-3 lectures, what we will do is try to learn principles of several techniques. First thing is about there are three ways of analysis we should do. One is about at the level of DNA, right? What is there any change in the gene or on the genome? Is there any kind of variation, right? Like mutations, for example. That is one level of study. Then we have another level of study is whether this gene is expressed or not. How is it expressed? How much is it expressed? When is it expressed? Those kinds of things. We'll have another set of techniques to analyze at the level of transcription, also at the level of translation. And then we also have one more that's not mentioned here is analysis of gene function. Right? So if an RNA or a protein is there, how can we determine its function? Does it interact with another protein? Does it interact with RNA or or DNA? Some kinds of techniques like that. So each of this is a category. And in today's class, we will try to see some techniques that are analysis of genetic variation. Please bear in mind that we already have done some of these techniques. It is a good way of reviewing as well. And most importantly, the lecture is supposed to go as a kind of discussion. Okay, you will have questions. You should try to answer them as well. Unmute and try to answer. So in genetic variations, when we are reading, we have multiple levels, right? We already discussed these, but it's a good review also. For those that may have missed, it's a nice way of trying to recollect and review what we have done, we have studied so far. There might be point mutations or small scale. That could be substitutions. Substitutions again can be of two types. You may have transitions or transversion, right? Transition is purine to purine or pyrimidine to pyrimidine, right? When there are changes between the bases, then you call it substitution. One is replaced with a different one. Transition, if a purine is replaced with another purine, substituted with another purine, or pyrimidine is substituted with another pyrimidine. It is transversion is when you have a purine, something like this. You can have a pyrimidine replaced with. You can also have a purine replaced with pyrimidine, or a pyrimidine replaced with purine. Those kinds of substitutions come under transversion. There could also be insertions. Here, when we say insertions, we are talking about small scale, 1 or 2 bases or 3 bases, something like that. Same is the case with deletions. We are not talking at the level of genetic information. We are talking at the level of the DNA. Okay, genetic information is DNA is just like as a physical structure. We are talking about it as DNA. DNA, the sequence of the bases is the information, right? That information is used to generate, to produce functional RNA or proteins that actually build the rest of the cell, right? So the changes in the DNA can influence the information. And those mutations, we have a separate category like silent mutations, missense mutations, or frame shift mutations, and so on. We are not discussing that at this stage. And the large scale changes can be insertions or deletions, which are large, maybe tens or hundreds of bases. There could be inversions. Inversions, the size remains the same, right? Something like B. In the last class, we also described, we did site-specific recombination, and there we have discussed about the Cree-Loxby system. And if we had a system like this, where the recombination sites are in the inverted orientation, then they are likely to undergo inversions. So A, B, C, D, E, the genes, and if inversion has occurred, it would have become something like this, right? So that is an inversion. The overall size remains same, but the inversion has happened, and we should be able to figure that out. And duplications may happen, and also transposition may happen. So these are large scale changes. And our job is to employ one or more techniques to find out or to be able to determine the nature of genetic variations. You can always add sequencing to all these things as well. Okay. So I wanted to have, yeah. So as a part of genetic variation, we will learn a few techniques. There are almost, they are the ones that predominantly used, popular ones. We have already finished Southern blotting and RFLP also, restriction fragment length polymorphism. We have, PCR based methods, we have already finished, but maybe we'll see if we have to do. Variable number tandem repeats, that is one more thing we need to see. Sequencing, we have finished. Allele specific oligonucleotide probes, we have finished. SSCP, single strand conformity, we haven't done this. We will, polymorphism, sorry. We haven't done, we came across it, and I told that I will describe this at a later stage. So that is what we will also look into. And these are specific to a locus. That is, if we already know or kind of we are interested in particular gene locus or a locus within the chromosome, that my lab is, for example, working on this. I am interested in that. And I'm trying to look specifically what may have happened in this particular genetic locus. There are other high throughput techniques like genome-wide comparisons. It's like saying, I have two bacterial cells A and B, strains, assume. I want to know what genes are present in this and what genes are present specifically what are present in A and specifically what are present in B. I'm comparing the whole genome, right? And those come under genome-wide comparisons. So here is the first thing. Until now, I think I already described the way you have to remember is for each of the technique, you should know what is the objective. When do we need such kind of things, right? Say, for example, Western blotting. If the application, what you should remember as a Southern blotting or Western blotting, it is about, Western blotting is about whether a protein is present or not. Yes, protein is present or not. That is the actual core objective of this technique. And the principle depends on this particular objective, right? Whether the gene is present or not, all those things will come later. First, the actual essence of Western blotting is to find out if a protein is present or not. Upon that, if we have done the semi-quantitative way of doing the Western blotting, then we can probably figure out how much of this protein is expressed relative to a standard. Okay. And one should also know the principle, why, what kind of principle it is. In Western blotting, for example, we use blotting technique, electrophoresis blotting, then primary antibody, secondary antibody, and so on. And we also have a blocking step and so on. So try to know all those stuff. We should also know the limitations, what it can be used for and what it cannot be used for, or what it can say and what it cannot say. Okay, so those are some of the things. Say, for example, in Western blotting, if you had, there are two samples, A and B, two bacterial cells, and we did protein Western blotting for a protein called XYZ, and strain A does not give a band, whereas strain B has given a band. What can you say? Can you say that there is gene XYZ is absent in A? Can you say about gene XYZ? You cannot say anything about it. Can you say that transcription of XYZ did not happen in A? You cannot say that. All you can say is protein XYZ is absent in the sample. Okay, sample or sample of A in that particular growth conditions. So that is how one has to be very clear about what you are speaking about. In research, if you see the olden or say 50s and 60s, or maybe prior also, I have gone to 50s papers sometimes. I like their style of doing it. Of course, for today's standard, it's not too much, in the sense in today's standard we do a lot of genome-wide and high throughput ways. But if you see the 50s and others papers, you will find the sincerity of the scientist in the research article. They would not say any more or less than what is seen. Right? That is the code, I think, everyone who wants to be in science should follow. You should not say more than what you can see in your results. You should not say less than what you see in your results. Sometimes we get negative results. That means we tried out a hypothesis and say we get the hypothesis is wrong, and then we move on to something else. In today's papers, in majority of the time, it is about promotion of the research work rather than the science itself. So in olden papers, you might find occasionally a sentence saying, we tried out this particular technique for this, but somehow we could not get any result. That's a good way of, sincere way of doing science. So one should be able to follow these kinds of attitudes when one wants to be a scientist. We are in current day, we are much more in competition in the name of about the number of papers and so on. Okay, it's rather than the quality of the sometimes it compromises the quality of the research. And as youngsters, I would always I would try to tell everyone, anybody wants to be in research, you have to be you have to maintain utmost integrity, so that in the sense that there should not be any kind of fabrication or miscarriage of information or the science and results. We should be utmost sincere. And that is when we are generating quality knowledge that could be useful to many other people in the future. Yes, and then you should also know about the applications of the technique. One important one I didn't mention here is the title of the technique. Okay, each of the technique has a name, and you should be able to associate the principle with the title. If you noticed or not, I try to talk in terms of, say here, for example, I was talking about Western blotting. When I was talking about blotting techniques in general, I described what exactly blotting means. I think I told something like, we spilled some water or something. We take blotting paper, which is a paper usually available in the labs, to put it on. That blotting paper will absorb all the spilled chemical or liquid, and then you throw it in the dustbin. That's a blotting paper. I don't know if you have used it before. So blotting is trying to pick up something using by say by absorbing using a sponge, for example. You're picking up something that is spilled also. In southern blotting or western blotting, you have it in the gel electrophoresis. You have the DNA. You're putting in nitrocellulose membrane and trying to pick up the DNA molecules. That is blotting. So what I want you to know is try to associate the principle with the title. So anytime, because people are only going to ask you based on most of the times they know this particular technique, they're going to ask you with the name of the title. And if you're already associated with the principle, then it's easier for you to remember. Okay, try to do that. Because in the next 3-4 classes, maybe we will read about some 20 or 30 techniques. So keep that in mind. Here is a GIF of pulse field gel electrophoresis that we haven't discussed. We should have done, but I was waiting for this particular animation. I can't do that in the other software. So here is pulse field gel electrophoresis. You must have learned this in the analytical techniques course. So mostly when you are doing large DNA fragments, if you're doing, I hope you remember that the DNA size of the DNA size, if it is increasing, then if you should decrease the concentration of agarose, isn't it? Otherwise, the DNA will not be able to move through the agarose in this direction, right? So, but if you are trying to, if the DNA is very large, something like a genome, 3 or 4 MB or so on, then you have to reduce the concentration a lot. If you reduce the concentration a lot, because during electrophoresis heat is also generated, the agarose will melt. If it melts, then whole of agarose electrophoresis is gone. You're unable to do it. So then people have tried to come up with various techniques wherein one can do one can perform electrophoresis without reducing the concentration of agarose. So what was done is using pulse field gel electrophoresis. This is one negative electrode and this is positive electrode. So I will call it as A and A. And there is another one that is a negative electrode, B, a negative set B. And you have C, I'll call this is also negative electrode, and this would be positive electrode, sorry. So this is about alternating currents, electric field. You're applying voltage in different directions. So usually in normal electrophoresis, you would have one electrode here, negative, and here, positive. There are wells here into which you would have applied or put in the sample, and the sample will run in one direction. Whereas in the pulse field gel electrophoresis, there are multiple, at least in the shown diagram, there are 3 different sets. At any given time, you can have a set of electrodes active, then the DNA is going to move in this direction. If B is, after some time, the computer also can turn off a set of electrodes and activate B set. So now the DNA is going to move in this direction. And after a minute, after some time, it will stop it will disconnect the B electrode and electrodes and turn on C electrodes. So the DNA will move like this. And this will keep on happening, because of which, even though DNA is long, it will find opportunities or gaps to move across the electrophoresis, the agarose gel. Okay, so that is pulsed field gel electrophoresis. When you say pulse field, this is one pulse. When you activate A, that is one pulse. When you activate, you shut it down, and then you are activating B, then that is another pulse. You're changing the pulse of the field in a way. Pulse, you're having three different orientations of pulses. You can think of it that way. Okay, there are several variations. Sometimes there are also oppositely placed electrodes also, but I hope you get the point. This is used, application is for electrophoresis of large fragments. Okay, and the necessity is because if you reduce the agarose concentration too low, then the agarose gel would melt. So to circumvent this, alternating currents in three different sets or sometimes multiple of those can be used to allow the electrophoretic separation of large DNA fragments. So if there is if you can, for example, I'm trying to tell about an application. You can take, I have three different bacteria. I have E. coli, and I have three different strains, A, B, and C. I want to know what is the difference between A, B, C at the genome level. Are their sizes equal or different? And in that case, I can do pulse field gel electrophoresis, and I can figure out, I can see if there are if there are variations or not. If they're all similar, they would travel in the same lane, okay, same size. But if they have some differences, they're likely to have different sizes. At least that can be a good enough technique, analytical technique, to find out if they are different or not. So here we are going to restriction fragment length polymorphism. I'm going to give you it. This is the gel, A, B, sorry, 1, 2, 3, and 4. I want you to tell me what can, say, assume A as normal. What is restriction fragment length polymorphism? You take DNA and you digest it with one restriction enzyme, and then it might find fragments like this, right? When you run the electrophoresis, it might give a band. Okay, this is basically with southern blotting, right? So you know, if I have used a probe here, right, only that probe will give a signal. Only that fragment will give signal. So I have taken from the same species, except there are different strains, like in E. coli, for example. And I took DNA from 4 different strains, 1, 2, 3, 4. I performed restriction digestion, and then I performed southern blotting for one particular gene. Okay, so what? Assume 1 is normal. What can you tell about 2, 3, and 4? S. What can be assumed? 2 could be it has, if it is having higher length. What does that mean? Okay, this one also has 2 and 3 have fragment that is larger than that of the normal one, right? So what does that mean? There are more than one possibilities. I think I did describe in several of the techniques. It's just an analytical technique. Can you answer, anyone? 2, 3, and 4. What could be the reasons? Okay. So the reason could be something like this, right? Here is 1. I said this is the fragment detected because probe is homologous to that region. In 2 and 3, they both are giving larger size fragments, and there could be 2 reasons at least. In one of the cases, it could be a loss of restriction site. Say if a point mutation happened, And what was GAATTC? Okay, is mutated to GGATTC. It is not EcoR1 site, because of which EcoR1 is unable to cut it here anymore. So the whole fragment would be detected, because of which the size has increased. Is there any insertion? There is no insertion in this. Yet you are seeing a larger fragment. One of the possibilities is the loss of a restriction site. That is one way of thinking about it. But there are two possibilities, as I was saying. Either it could be a loss of restriction site, or it could be an insertion. That is also a possibility, like in the case shown here for 3. In 4, sorry, it could be a deletion. Like in the case of 2, it could be also a generation of a restriction site. That is also a possibility. Okay. Say, for example, a new restriction site has formed here because of a point mutation. Now the fragment will be smaller, isn't it? So if you are in fragment length polymorphism, restriction fragment length polymorphism, doing through southern blotting, if you are seeing an increased length, it could mean loss of restriction site, or it could be that there is an insertion, right? I hope you remember where we have done with epigenetics, methylation sensitivity and all. There also, it is not about loss of restriction site, but there it is about methylation of the restriction site or not. Here, it could be a large-scale mutation or a small-scale mutation. Small-scale mutation might be because of loss of restriction site or generation of a restriction site. Large-scale mutation could be insertions or deletions. Okay. So again, the technique name RFLP, restriction fragment. You're doing restriction digestion and fragment length. So from here to here, this is the restriction fragment that we are looking at, and we are seeing polymorphism, right? When you say polymorphism, it would mean that in some cases, this is assume normal. Polymorphism means many forms. Polymorphism, many forms. Okay. So the same fragment in different strains is having different forms. In one case, it is much larger. In some cases, it is smaller. In larger too, because of insertion, of course. And it is, in some cases, it is smaller. Okay. Restriction, fragment, length, polymorphism. Okay. And here is another one. It is also about restriction, fragment, length, polymorphism, but it is about transposition. Here, I hope you remember about transposons. Transposons are jumping genes. There are two ways of how it can happen. One is cut-paste method. It can get cut from here and move into a new place. The other one is copy-paste method, where the place it has transposed would be retained, whereas somewhere else in the genome, it would copy itself there. Copy-paste methods. So if we are going to use to analyze transposition, and you have made a probe against the transposon, if it gives one band, what does that mean? And if it gives an additional band, what does it mean? How many copies do you expect? So in the first one, you would have expected only one copy, like this. IS is for insertion sequence. And if it has moved to somewhere else, then you will have an additional copy. And it will be detected because you have chosen a probe that is specific to the transposon sequences. Okay. So this is again restriction fragment length polymorphism for studying transposons. How many copies of this transposon is presented in this gene? That's how one can study. Here, variable number tandem repeats. Okay. Okay, so tandem repeats are something like this. You have some 15 or 20 bases. Okay, and in the genome, in our genome, we have a large number of copies of these. So this sequence is the same as identical as this sequence. Okay, when you say tandem, that means if you have multiple copies like this, that those are tandem, right? And they are the same repeats. So you have tandem repeats in tandem. It's something like concatamers when we were learning about lambda phage replication or formation of new lambda virions. So why do we use the word here variable number? Because in some individuals, you may have these 10 repeats in tandem. In some cases, you may have just 3 repeats. Okay. In some cases, you may have 20 repeats. So that's why variable number tandem repeats, VNTR. Our genomes have majority of the genomes have large number of variable number tandem repeats. So if we are going to do, you need to understand this experimental setup. If we already know where these tandem repeats are occurring, say these are the tandem repeats, and this is where the first one starts, and this is where the last one is. If we should then be making a primer on the non-repeating sequence. This region is not a part of the repeats. Okay, whereas these regions are whereas the middle ones are the tandem repeats. This one has 1 2 3 4 5 6 7 times 7 repeats in tandem. So we should have designed primers like this, and then if we have done PCR, then if it has 3 copies, it is showing a band like this. If a B has more number of copies, so this total size from here to here is more, because of which the PCR fragment or the you call it as amplicon. Okay, amplicon is the DNA that we are trying to amplify, or the fragment length that is formed after amplification. So the amplicon size would be more because there are more number of repeats compared to that of A. And in C, it has even more number of copies. So in several typing methods, something like RAPD also may be having these kinds of things, where if you can try to find out number of times it is repeated, and you can find a relationship of inheritance from one to the other. They can be used. Okay, I hope you understood this variable number, because in different organisms you have numbers are different, and these are we are analyzing the tandem repeats that are present. Tandem is continuously a one repeat being present continuously one after another. Yeah, I have to discuss one thing so that I want to see if your understanding is good or not. If I had made a primer here and here, what will be the amplicon size in A, B, and C? Right? I'm having primers specific to the tandem repeat. So it's not, it is too small for that probably, but still. If I made primers for the tandem repeat like this, okay, what will be the size? What will be the size of the amplicon? It's likely that if it is binding here, it can also bind here and here and so on. And if this one could give one product, this one could give one product, and this one could give one product. All the products are of the same size, and they are very small. And sometimes you may have this primer and this primer giving a product. So you may have a different length of different size of the fragment. Sometimes you may have this primer and the terminal last primer giving, and then you may have a larger one. Now we have a confusion, right? Is it this one is this correct? Is this correct, or is this correct? Of course, logically, by the experimental setup, simplistically, by seeing from here, we can assume this is correct. For example, if this in form, then does it mean that the A did not have 3 copies? We cannot say. That's why we should actually always have the primers on the non-repeat, the border genes to the repeating regions. These are the tandem repeats. You should take primers that are only represented once. Then you can get the total length of the tandem repeats. Okay. Yeah, single strand conformation polymorphism. So this is about we all know that RNA is single stranded and so on. Yes, it is usually single stranded, but it does fold upon itself. Intramolecular hydrogen bonding may happen, right? You know about the hairpins, like the tRNA molecule, for example. It folds like this, forming several hairpins, like something shown like this. So, right, it's easier to remember this way. So the base pairing between the intramolecular base pairing is dependent on the sequence, right? If there are any kind of mutations, especially those that are a little larger, the same thing, it might not form the exact structure. It might form a little bit varied structure, right? So what happens because of it is the normal one may have one kind of conformation, whereas the one with the mutation may have a different kind of conformation. So look at the title of it. Single strand. They are taking one mRNA, one strand of mRNA. And we are looking at conformation polymorphism. This is one conformation. This is another conformation. If we have another RNA, same RNA from a different organism, which may have a different mutation, it might form something else, some other structure. Okay? So you have polymorphisms in the conformations of the single strand RNA of particular gene or so. So how we can separate these is based on the structure, the compactness of the structure, they will be able to move down the gel. If they are much more compactly packed, they will travel faster. If they are loose in conformation, then they would travel slower. That's so. I hope you remember from plasmids, the supercoiled one travels much faster because it's much more compact compared to that of linear or relaxed plasmid. So here are two examples where we are supposed to do it in RNA, okay, but we don't have to do it only with RNA. You can also do it with DNA. But the point is, one has to generate a single strand DNA. Okay, so what people do is they use a radio labeled end label. I hope you remember end labeling. End labeling, the substrates are gamma labeled ATP, and the enzyme is polynucleotidyl kinase. So you have end labeled a primer, and you allowed PCR for a normal one and say a mutated one. And then you have generated the PCR, and you would have denatured it. And then you allow rapid cooling. Here the cooling should be rapid. If you allow slow cooling, it might find its complementary, some other complementary strand, and might form double-stranded DNA. But if you quickly cool it, then it will more preferably try to find base pairs within itself and have intramolecular base pairing. So it forms a certain structure based on the stability of the the hydrogen bonds and so on. So each of them will have their own conformational states. And when we run electrophoresis, assume that this one is traveling much slower compared to this one. You will not be able to detect the other strand because we haven't labeled the other strand anywhere. We have only labeled one of the primers. Then now we know that based on looking at the gels, you have seen that one is traveling slower compared to the other one. So all you can say in this is there is a mutation. There is a difference between the sample A and sample B. In terms of that part of the DNA, there is a difference. You cannot say more than that. You cannot say if there is a substitution or all these things. You can only say that there is some change. Here is how the SSCP results look like. The technique is almost same. Okay, you would see some, because you would see strands bands like this, and that will indicate that there are differences between, say, A and B, C, D. You can say that there are differences between them. That means there might be small scale mutations, which could be insertions or insertions, deletions, or substitutions. That is called as single strand conformation polymorphism. You are doing it for a single strand of DNA or RNA. Preferably, it is mostly done in DNA, because DNA is much more stable. RNA is difficult to work with. And we are looking at conformational polymorphism. And here, conformation is based on intramolecular hydrogen bonding. So now we will look at genome-wide comparisons, and I will look only one, and then I think we will stop there. It will take a lot of time. So genome-wide, in the last 30, 40 years, or say 30 years or 20 years, everything has become high throughput. Nobody is looking at individual genes anymore. We are now trying to do in one go, try to find all the differences between one cell type to another cell type, or one strain to another strain. That means we are generating more amount of data, and then probably from there on you can pick up, and different individuals can pick up and work on the smaller level. I hope you know holism, right? Holistic approach and reductionist approach. Right? Everything you can think of. This is assumed genome, each consisting of several genes, right? For a long time, different people say one research team worked on, 3 or 4 research teams worked on gene B, and the other ones worked on gene C, and like that. That means it will take a lot of time to find out what is happening in total. High throughput ones, now you can analyze at the level of gene, you can analyze at the level of transcriptome or the mRNA. This is at the genome, and you can do it at the proteome level as well as, and now we also have a little bit extension to this metabolome and so on. So that means your actually all this OME is added from the genome part, I think. When you're trying to do all of it, then the transcriptome is about analyzing all the RNA that is ever transcribed. Proteome is all the proteins that were present in a cell, and metabolome is all the metabolic molecules, metabolites that are present in a cell. So here everything is based on its. You cannot see what is happening actually. There is a lot of dependence on the computers. And now we have there are small chips which can measure a lot many more things of what can happen, whether the genome is present, whether a particular gene is present in a genome or not. So the microarray technology is something like this. This is the chip, and when you say array, what is an array? You have several sets of these, then you call that an array of something, right? And here we have array of spots, and it is very small array. That's why you call it as microarray. Okay, that means you are having large number of these. Here we have spots, and each spot, okay, here it is enlarged, is having single-stranded oligonucleotides, and they are with a specific sequence. So each spot has multiple oligos, or larger, a little more larger, but all of them will have the same sequence, right? You have 1, 2, 3, 4. All there are 4 single-strand DNA molecules at this spot, and all of them have the same sequence. So the computer knows in each of the spot what kind of sequence is there. Okay, and like that you have so many of these, and each gene is represented, typically each gene is represented by a spot. One gene is represented by one spot. Like that, almost all the genes of protein coding genes of a human genome, something like approximately 25,000, or say 20,000 genes is provided on this one chip that is somewhere near the size of a thumb or so. But the printing of these and making the chip, everything is automated and done by robots. And people use several strategies of having to, if you want to compare two different samples, A and B, for example, then all the genes or DNA fragments that you are looking at, you can make it into smaller pieces, and all of them could be tagged with something like one fluorophore, right? The same thing can be done for B with a different color, like red. And in some spots, you may have only the red one hybridizing. As I told, this is the DNA that is stuck to the spot, to the glass slide, and it is single-stranded, and it has a specific sequence. And if we have, when we add this sample here, and if the sample has a complementary sequence to it, it will base pair and stick to it, and that has a fluorophore with red in color or a green in color. Sorry. If a spot is giving only red color, what would that mean? That means only B has this particular gene, but A may not have it. And in this case, in the second one here, only green color, which would mean that only A has that particular gene, not B. And for this gene, both green and red are there, which would mean that A as well as this gene is common in A as well as B. That is what the implied meaning is. And if we see that red and green are together, then the light will come as mixed, and which would usually be yellow in color. Okay, so different people use different thing, but red and green are popular, I guess. So the overall, I will stop with this one slide, okay? So you can do many things, but I'm looking at oligonucleotides, not the cDNA. Because I'm trying to, here I'm trying to read at the level of genetic variation. That means I haven't come to expression or genetic gene expression. If I'm doing cDNA, then I would be working at gene expression level, because cDNA is made from mRNA. Here, the objective would be, because based on what I'm trying to teach today, I am trying to look at two different samples, and I want to know what is the difference, genetic variations on the DNA that are there in A and B. So for that, we will, first thing is to prepare slides. You have to make oligonucleotides, and each gene represented by one type of oligonucleotide. And they should be spotted onto glass. For all, gene 1 should be, the oligonucleotide should be spotted here and fixed to the glass slide. For gene 2, all the oligonucleotides will be, they have all the nucleotides in this place, in this spot, will have the same sequence. And like that, you should have, one should have made the slide or the chip, which is the micro array, right? You are making an array of spots, each spot containing identical oligos, and those oligos are actually complementary to the genes. Each gene in each spot contains oligos that are complementary to a particular gene in a genome. When we are doing experiments, this is the experiment part. We might take control cells or healthy cells or a different type of organism also. It depends on what you're trying to compare. You can compare two different organisms or strains. You can compare A, uh, A from one location and A from a different location. There will be some genetic variation between the same organism or species, but they might have some mutations or changes. So here, based on the experimental setup here, you can take a healthy cells and assume cancer cells, right? DNA, or this is for transcriptomics. I'm not going to that. You can take the DNA molecules, cut it down into smaller fragments, and then label them. So the healthy ones can be labeled with red, or green ones can be, sorry, the diseased one could be labeled with green. And then we would label, then we will mix it up and then hybridize to the slide. This slide, we cannot make anything. They're very small, and about 20,000 genes are there. So it is usually scanned by a machine, which is with under a microscope. It has all the information, which spot has which gene, right? And then all these spots will look something like this. I think I may have different pictures somewhere. If not, I'll put it for the next class. So it will form a complex pattern based on red, green, or yellow. And then you can find out, the computer will tell you which all genes are specific to this condition, and which are specific to the diseased condition. That is how you can figure out which genes are present or not from a particular between two, when you're comparing the genomes of two organisms. The same technique also can be used for transcriptomics. If you're using cDNA, if you're taking cDNA from two different or mRNA from two different cells, that would be transcriptomics. Okay. Yes, I'll stop here. In the next class, we will discuss about gene expression. But otherwise, if you have any questions, we have 2 or 5 minutes you can take. Please let me know if you have a question. Hello? Anybody?